Welcome to Data in Biotech, a podcast from Cordine where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they're using data science to solve technical challenges, streamline operations, and further innovation in their business. This week, we sat down with Mike Nally, CEO of Biomedicines, a company that is revolutionizing the way medicines are created by using a new approach called generative biology. During the interview, we discussed the use of generative AI-based approaches to construct new proteins and solve the challenges of declining research productivity and drug pricing in the pharmaceutical industry. Mike also introduced us to Chroma, an open-source diffusion model that expands the natural universe of proteins, and shared his thoughts around the feedback loop between computation and experimentation in protein design. Finally, we touched on Biomedicines' culture, one that emphasizes audacious ambition, being intrinsically digital, and patient centricity. Here we go.
Mike Nally, welcome to the Data in Biotech podcast.
Thanks so much, Ross. It's great to be here.
Awesome. Just to kick us off, would you give us just a brief introduction to you and your background and what brings you here?
Yeah, so I'm Mike Nally. I'm the CEO of Biomedicines and a CEO partner at Flagship Pioneering. I joined both Generate and Flagship a little over 3 years ago after having spent 18 years at Merck, worked across most parts of drug discovery, development, commercialization, and was fortunate enough to see what I thought was a transcendent technology a few years ago where Generate was using generative AI-based approaches to construct new proteins that nature hasn't been able to discover. And that belief has only grown over the past 3 years.
Yeah. So what excited you about Generate Biomedicines that made you want to step out of Merck and take on this new challenge?
Yeah, it was one of those things where, over the couple of decades that I was in the industry, I stepped back and said, what are the things that are actually holding back progress? And one was this troubling trend on research productivity, where if you look at it in the aggregate over the past 4 decades, we've seen declining productivity consistently at an industry-wide level. And secondarily, you had this kind of somewhat interrelated, but oftentimes independent dynamic where pricing practices from the industry led to inadequate patient access and ultimately undermined the reputation of the industry. And a lot of my career has been oriented toward how do you solve these kind of two monumental challenges. And I became a clear believer that over time, with the use of technology, we'd be able to understand biology in more profound ways than the human mind alone could understand it. While we've had some of the best human minds on the planet studying biology, we still only know probably about 10% of it. And if you think about how humbling that is, we've been staring at it for all of our existence, and yet we know so little. Could this era of technology help us understand it in more profound ways? And I became a big believer that this intersection would be the path to reverse those trends that I talked about earlier, that you could reverse the productivity trend. And then by reversing the productivity trend, you could rethink the pharmaceutical business model. And when I saw the early data at Generate, I almost fell off my chair. I had never seen a technology that held the promise. Now, I will say when I first met the team here about 3.5 years ago, it was still very early days. The data was relatively scarce. But what became very quickly apparent was that if it were to hold true, it would change the way every drug is made in the future. And that's what's been so exciting about it for me.
Yeah, that makes a lot of sense. So can you tell us more about Generate Biomedicines and why you think that this company is well positioned to solve the R&D productivity problem as well as the drug pricing problem?
Well, so if you take it at the first principles, right? I mean, drug discovery has been an artisanal craft. So it's down to an individual genius, a team of geniuses that have a fundamental biological insight. And then basically we borrow from nature and we tinker with this in a trial and error process to try and get a therapeutic. And it's no wonder why this is such a difficult business, right? I mean, it's easier to put a person on the moon than to discover a drug. And, what we also know, and this has been the fascinating journey I think we've been on as an industry, is that when human beings are conceived, what's passed from parent to child is a code, DNA. And yet that code then leads to all biological function. The problem is we've never understood the code. And, so this is truly a coding problem. And, I believe biology is the first information technology, the original information technology. We just have, until about, 70 years ago, we never understood. We couldn't see the code. We could never read the code, yet let alone program it. And I think with the advent of new tools and technologies, we're now at a point where programmability starts to become a distinct prospect. And we're very early in that journey. But if you think about the starting point, understanding about 5-10% of biology, if we could double that over the course of the next decade, the amount of good we could do for humanity, the amount of disease we could cure, would be monumental. And so for me, that's the exciting journey that we're on. And, in almost a simple way, whenever these sort of complex fields become engineerable, industrial revolutions occur. We've seen this with chemical systems. We've seen this with electrical systems. We saw when, Bernoulli distilled the principles of flight down to a mathematical equation, all of a sudden the aerospace revolution takes place. Biology is on that precipice. And I think that's what's so exciting about the field, about where we are, about the work that Generate's trying to do, is how do we distill that down as quickly as possible to have that sort of monumental impact?
Yeah, that makes a lot of sense, and it is very exciting. It sounds like just from the name Generate, that between the reading of the code and the writing of the code, that sort of the differentiating feature of Generate is that your ability to write the code. Am I thinking about that right?
You are. So the rate limiter for us in many respects has been data, right? What we've seen over the past decade is a convergence of three major themes, right? We saw a massive increase in computing power. And we see, even with the news last night of these blowout earnings from NVIDIA, the demand for compute continues to grow pretty massively. The second theme has been advances in machine learning techniques. And some of the core techniques that we use at Generate have basically been invented in the past 10, 15 years. And so you have those two dynamics at play. But with biology, unlike the advances we can make in machine learning in large language models, we don't have the internet to digest. Data is much more disparate, and Generate was set up basically as an integrated dry and wet lab. And I think this is a really important piece. So every DNA sequence that our models will spit out, we build into the protein format of choice. We test and we measure. And then we have an informatics layer that sits on top where every data point is captured and fed back to refine the computational approach. Now, when you start a company like ours, you're reliant on external data, and proteins had two extraordinary data sets. So they had the protein data bank, which gave us about 200,000 high quality, high resolution structures of proteins. And then we had the sequencing revolution, which gave us 180 million amino acid sequences across all species. That allowed us to start to understand interrelationships between sequence and structure. But if you're interested in therapeutics, what you really care about is function. And so our wet lab has been our attempt to classify and quantify structure, or function, I should say, in a much more profound way. And we've now been able to generate millions of proteins that nature hasn't discovered. We've built all those, we've tested all those, we've learned on all those. And the models are consistently improving, giving us better and better answers, which is what I think gives us so much hope that this will be a pretty profound revolution to address that productivity equation. And then if we're more productive, we don't have to be reliant on traditional pricing models. We can think differently about how you price things. And I can give you some specific examples as we continue the conversation, certainly.
Yeah, no, this is really interesting, and I love the way that you characterize sort of the feedback loops between the dry lab and the wet lab, and the way that the data is being fed back into the system. Can you help us think through the layers of modeling that are occurring, and how sort of the broad space of proteins get searched, and then how you decide which ones you want to experiment with, which data points you want to gather, and then how you feed those back into the system as a whole?
Yeah, so it's a great question. Traditional antibody discovery is done by immunizing a human, a mouse, or a llama. We then cull through plasma, we see what sticks to a target, and then we try and modify it to have drug-like properties. Generates models start with structural information. So do we have the structure of a protein binding a target of interest? Or in the kind of most extreme version, do we just know that there's a target that we want to interact with, but we have no prior knowledge of anything ever binding to that target? That second example has been the holy grail for the protein design field. It's called de novo generation. And as far as we were aware, and we are aware, that had never been done before 3 years ago. On the first example, where you have this co-crystal structure of a protein binding a target, if we ask our models to make suggestions, so suggest other DNA sequences that perform as well, if not better, than the reference molecule, our hit rate right now is about 60% straight out of the computer. So 6 out of every 10 will be highly functional molecules. And what's been really interesting is the diversity of those molecules is extraordinary. And when I say diversity, if we look at the sequence that leads to the reference molecule, and then we look at the sequences that come out of the model, we can change 30, 40, 50, 60% of that code while retaining or enhancing function. Historically, if you think about protein design, the way we've designed proteins or the way we've manipulated proteins is either we use random mutagenesis. So we do something called error-prone PCR, where we will mutate the protein sequence, or we've historically used computational techniques, biophysical-based methods like Rosetta. This is a software-based approach. Using those sort of approaches, you can change about 10% of the binding region before mathematically you find no functional variants. And this is just simply because the search space for proteins is almost unimaginably large. If you think about the average protein being, let's say, 200 amino acids, and there are 20 natural amino acids, the combinatorial possibilities are 20 to the 200th power. That's atoms in the universe square or cubed combinatorial possibilities. That is just an array of opportunities that are really hard. So having good prior models that allow us to search that space really efficiently is at the core of what we do as a company. This concept of de novo generation now, and this is what I think gives us the most excitement. We saw our first de novo antibody hit 3 years ago, and that had been structurally confirmed. De novo generation gives you is control over specificity. So if you think about the randomness of relying on the human immune system, a mouse's immune system, to generate antibodies that bind with specificity to a given location, we have no control over where those antibodies bind. They bind where they bind. We estimate that roughly half of the target landscape is inaccessible. These are like immune cryptic domains that our bodies don't generate antibodies to. And if we have now tools that can give us those specific answers, that gives us tools to prosecute biology in ways that we've never had before at our disposal. And we focus in on both of those technologies. We focus in on an optimization technology where we can co-optimize things for all of the relevant therapeutic properties in one go rather than relying on sequential processing. And that allows us just to get better answers faster than traditional techniques.
Yeah, I love this. And maybe this is a place where we lead into Chroma and the open source model that you've released. Because what I noticed about Chroma is the level of control that you're given over the input that you give to the system in order to guide the development of the de novo proteins. Maybe I'm an outsider, but would love to hear your introduction to Chroma and how you view the way that model feeds into the generate model that you're discussing about.
Yeah. When we think about Chroma as one of dozens of models that we use, what's unique about Chroma was the first diffusion model publicly released that allows us to expand the natural universe of proteins with extraordinary precision. If you think about the 3.5 billion years the planet has been around, nature has basically given us about 20,000 unique protein concepts. We believe that is one drop of water in a bucket of potential proteins. And what Chroma, and you can see some of the examples behind me, gives us are mathematically correct proteins that when we put these codes together, they perform and fold like the proteins that nature's discovered. And again, this goes back to what we were talking about earlier, Ross, where if you think about the search space that we're working in, sometimes Mother Nature is an extraordinary technician, and evolution is an extraordinarily powerful tool. But given the vastness of the search space, we estimate that evolution has basically touched one drop of water in all the Earth's oceans of potential proteins. What these sort of tools are giving us is an ability to survey the oceans. And inevitably, when you survey the oceans, you're going to find some pretty cool stuff. And so Chroma is one of the latest versions of it. What we do with Chroma at Generate is Chroma's trained on the publicly available data. We then have a series of conditioners that we apply to Chroma to take these protein concepts and turn them into therapeutics, purpose-built therapeutics for unmet medical needs. And it's this combination of both diffusion models like Chroma with a series of other models that allow us to dial in very precise therapeutic attributes that address major biological challenges.
Yeah, so what I've seen is that there's this kind of scaffolding that's going on where you're using the diffusion model to generate the candidates of the proteins that you might explore, but then you've also got this Bayesian optimization approach that's applying constraints to what might come out of it, but then also providing a methodology from which you learn from the experimental data that you're gathering over time. Am I thinking about that right? Or how does it look on the ground?
100%. And I think it's this virtuous cycle between computation and experimentation that's so powerful, right? One of the things that has been the Achilles heel of these generative-based approaches early on in many other domains are things like hallucinations. With models like Chroma, and when you're using experimental validation, hallucinations become features, not bugs. Because what that does is it allows us to refine the computation, recognizing the codes that don't work as much as the codes that do work. And for us, the exciting part is when we created this model, we had no idea what percentage of these proteins would express, what percentage would be performant. The rates have been well beyond our wildest dreams. And that basically tells us we're getting pretty good at understanding the underlying mathematics of what it means to be a protein. And in doing so, it should allow us to come up with new concepts to treat some of the world's greatest diseases. But also, the nice thing about these sort of protein-based models is therapeutics is just one application of them. We can think about if we can manipulate enzymes, can we make a more efficient version of Rubisco for photosynthesis? Could we think about entirely new surface materials replacing plastics? These sort of things have transformative potential across so many domains. We've just particularly focused on therapeutics because of the unmet medical need that could be addressed with it in its early days. But we think the potential of these sort of approaches is far wider.
Yeah, that makes a lot of sense. So you mentioned data earlier. Is data the main limiting factor for what prevents you from exploring even more of the space? Or is it how do you think about the potential for expanding into other areas and leveraging these capabilities that you're developing internally?
I think it's people, Ross, in all honesty. When I started at Generate, we estimated that the number of people that were skilled at the intersection of machine learning and protein engineering was about 90 people on the planet. Most of those folks were working for large tech companies, interestingly enough. And that part of what we're seeing is this blending of disciplines, right? So classical training, you were a computer scientist or you were a biologist. Blending those two was a rarity. And I think what we're trying to do within Generate are find people who are multilingual, right? Who can understand computation, understand experimentation, understand therapeutic development, and as really well, these are rather tribal cultures, right? The clinicians don't speak the same language as the computational scientists. And how you create a new vernacular for this kind of these unicorns that can span these different domains is actually, I think, the biggest challenge, but also the biggest opportunity, because a lot of times the greatest innovations that society sees occur in these white spaces between disciplines when you're able to fill those in. And I think this is a classic case of having to marry three pretty disparate disciplines to get to a place where you can get therapeutics that can make a big difference in the world.
How has your view of the role of machine learning in the R&D ecosystem at a biotech organization evolved? Because I would imagine you've had one perspective coming from Merck, and then that things have expanded over time.
So I think what plagues the space right now is some people are claiming it to be a panacea for everything. And it's not. It is an extraordinary tool that every drug company will need to deploy broadly. That requires a lot of careful thought and consideration, because you need to have a talent base that can embrace it. You need to have a culture where data is seen as a common asset, not a proprietary asset, which is a major cultural shift. And you need systems that support an end-to-end data strategy. At the same time, while I say it's not a panacea, I believe pretty firmly that all parts of the drug discovery and development continuum will be positively impacted by these sort of computational approaches at different rates, at different points in time. But I think these sort of tools will be deployed across the continuum to improve efficiency. One of the things we've been confronted with pretty recently was we entered the clinic with our first two molecules, right? Traditionally, therapeutic discovery would take 3 to 5 years for a protein therapeutic to reach clinical development. Our first program was 17 months from concept to clinic. So you can see a considerable savings there. Then you get to the clinical stage and you're sitting there saying, okay, let's just dissect the problem. We need to recruit patients into a study. Technology can help us do that. Patients are identifiable. If you go to the back end of a trial, you need to close a study out. That's a digitizable endeavor. And then how do you select patients? What are the inclusion and exclusion criteria? Are there biomarkers that can make the study more effective and efficient? All of these things, I think, are going to be positively impacted by technology. But we also have to recognize, as you move toward the clinic, you enter a much more highly regulated space. And you're going to need the concurrence of an important stakeholder like the FDA on some of these things. And being open-minded, being a digital native biotech company allows us to think about these problems at a foundational level, because ultimately what everyone in the biopharmaceutical industry is oriented toward, how do we get better medicines to patients faster? And if we can use these sort of technologies to do that in a safe and effective way, those are the types of things that have the prospect of changing the reputation, improving the reputation for the industry.
Yeah, I love a lot of what you said, and it just brings to mind the question of two questions, really. One is, where is the human factor most valuable to the process? Like, where does human attention need to be placed within this process? And then also, how do you improve the way that the collaboration occurs between the models, the tools, and the people whose attention needs to be on the process?
I think it's an important point, because there's this overt fear within society that these machines will replace humans. Our people, our employees, our scientists at Generate are the entire key to our success. It's the machine learning scientists who develop the models. So having great machine learning scientists that can develop the best models is what makes us distinctive. Having great and innovative experimentalists who can think about these questions through this different lens is what builds the technologies that allow us to go from 100 variants to 1,000,000 variants in a cycle. And then we need the clinicians who understand unmet medical need, the PhDs who understand biology at a really foundational level to come up with the insights and say, if we're going to direct this technology, let's direct it here because this will make the biggest difference in patients' lives. And so for us, these are tools that augment brilliant scientists. And it forces us to, in some ways, rethink workflows. It forces us to rethink the how, but the absolutely essential nature of extraordinary scientists is as strong here as anywhere I've ever been. And I don't see that changing, certainly in our lifetimes. Perhaps down the road, we can get to that place. The long-term vision at Generate is to ultimately be able to instantly generate the optimal therapeutic to address the greatest unmet medical needs. Let's just say that would be a great day for humanity, for society, for the world. We're a long way from there, but we need to keep that North Star because that's what we're driving toward. We're driving toward how do we make progress in climbing that sort of a Mount Everest goal.
Yeah, I love that. Can you share any examples of like how the workflows have evolved as the tools have improved between some of the brilliant people you have and the tools you've created?
I just mentioned, I mentioned earlier some of the work that has been recently done on cell-free expression. Basically, if you have computational models that spit out DNA sequences, then the first thing you have to do is build the proteins. And if we use traditional methodologies, that can be a few week process to just build the proteins. Cell-free takes that down to a 2 to 3 day window. So if you think about data velocity, changing out that sort of a technology gives us insight in much more rapid loops. Because really is when you're trying to, when you have a data strategy, part of it's the quantum of data, part of it's the quality of data, and the quantum is directly affected by the size of the experiment and the speed of the experiment. And we're constantly putting in new technologies all along these pathways. Another example, if we're screening, what sort of screening technologies can we do, not in a 96 well plate, which is the traditional experimental volume, but using microfluidic devices. So we can go from 96 experiments simultaneously to a few thousand. All of these sort of things require entirely new hardware, entirely new ingenuity. And as you change out each of these steps, you're disrupting the traditional workflow. And so part of what we have to operate almost like as a software company, where we're constantly innovating in the techniques because nobody makes these devices. And then where do you swap them in and swap them out as they become more industrialized and productionized? Because obviously quality still is of paramount importance.
Yeah, that makes a lot of sense. What do you think distinguishes? There's been a lot of AI, ML driven drug discovery companies that have been started over the last 5 years or so. And I'm just interested in how you view the main differentiators between Generate and the rest of the field that's doing this kind of work. You mentioned small molecules versus de novo proteins, protein engineering earlier, but any other differentiators that you see?
It's hard for me to speak on behalf of all the other organizations because I'm sure there's a lot of other great organizations. This is an extraordinarily talented group of scientists that are passionate about this specific challenge. And I feel extraordinarily fortunate to walk through the doors every day and work with people who are far smarter than me, far brighter than me, they're ingenious in their designs, they're fearless in pursuit of creating extraordinary medicines for patients. And I know that sounds very soft, but it's those sort of environments that change the world. And I give a ton of credit to the founders of Generate, the team at Flagship, Kevork and Molly, who have been core members of the leadership team. They created a culture where we basically embrace 6 core values. The first of one was something that was almost like, when I read it the first time, I was like, wow, most organizations would never say that. It was being audaciously ambitious. And the group of scientists here, what good looks like is changing the world. Being intrinsically digital, it was another one because we're taking on a scalability challenge, thinking about how we are fiercely united as a team, insatiable curiosity. These were the things that came to the fore. And then as we've migrated and matured as an organization, being patient-centric. And the culture here has been very distinguishing. And the world needs a lot of these companies to succeed. We hope to play a small role in that. And we think with the people, with the ambition, with the support that we have, the technology is going to make a big difference.
Yeah, I don't know if you've gotten exposed to the controversy around this, but there was a recent BCG article about the success of AI-discovered drugs. And then there was some backlash to the findings from, for example, Derek Lowe poking holes in the methodology that was used to assess that. And I'm just curious, since you're on the inside, what your viewpoint is on the success or lack thereof, or what this entire conversation gets wrong about the enterprise as a whole.
Listen, I welcome skeptics. Right? The reality is people don't have to believe. We just look at the data. We're seeing something different. And the longer people don't believe, the longer they don't pursue these sort of approaches. That's good for business. Right? I think to judge this, we're in a long lead time business. It takes 10 to 15 years to typically create a therapeutic. So how are we judging this at this point with any real thoughtfulness? The technologies of today can't be reflected in some respects in the assets that are in the clinic today. And we know, like, our core model that we did a lot of the foundational de novo work on, which was invented in the 2018 period. If you were trying to do that a decade ago, it would have taken 100 years to train. The molecules that we're judging this on were almost impossible to have created in the very recent past. And we've seen this change in large language models. I think what society knows best is the difference between the versions of ChatGPT. We're on an exponential curve. And if people want to sit there and say, oh, it's different in biology, this, that, and the other, that's fine. We'll see. We see that the answers that we're putting into the clinic, that we're starting to see in our pipeline, are simply different.
Are there any updates you can share on your two leading candidates, or any other exciting things you have on the horizon at Generate?
Yeah, we saw the early phase 1 data out of the first asset that we put into the clinic was a program that was initiated post the emergence of Omicron. If you recall, I think the world was pretty scared. We were in the midst of the pandemic, and all of a sudden we saw this mutant variant that jumped every fence. And it led us to sit there and say, is there anything that we could do differently than what the world has done in COVID? All of the antibodies that had been made historically targeted the receptor binding domain. This is the immune dominant region where if you have natural infection or vaccine-induced immunity, our bodies produce antibodies to this receptor binding domain, and they block the infection potentially. Unfortunately, the virus is a wily foe, and the virus mutates away from these sort of blockages. But there are domains on the spike protein that are extraordinarily well conserved. And despite the world's efforts in finding answers here, nobody, as far as we're aware, had put an antibody directed toward the stem helix peptide in the S2 domain of the spike protein into human testing, because the antibodies that were found through traditional means were not potent and were not broadly neutralizing. We used our technology to create a potent and broadly neutralizing antibody to that domain. We have the phase 1 data. It's extraordinarily well tolerated. It's safe, very low ADA rate, and it neutralizes all variants of concern that have been experienced to date, including other sarbecoviruses like SARS-CoV-1 and bat coronaviruses. This is an answer that was probably not very feasible using traditional techniques, because I think if it was, the world would have found something like this before. Our second program is an anti-TSLP antibody for severe asthma. We put that into phase 1 at the end of last year. The beautiful thing about that antibody is that it is a extraordinarily high affinity antibody. The current product that is in that class is dosed every 4 weeks. Our antibody, we believe, will be dosed every 6 months because it's about a 20-fold improvement in the binding affinity. And that 20-fold improvement in binding affinity leads to about a 5-fold improvement in potency and gives the prospect of extending the dosing interval to every 6 months. We go back to where we started in terms of we talk about how do you solve productivity? How do you solve affordability? If I only have to dose something every 6 months, the cost to the system, the cost to the patient is fundamentally different. That's what gives you the prospect of ultimately democratizing the availability of these drugs. Now, we've gone after areas of de-risk biology in the early days of Generate, but we think over time our ability to we don't want to take on risks for taking on risk's sake, but to give us tools to give us better answers to validated biology is something that's going to be exciting. But then ultimately taking on some of the great therapeutic challenges with these unique tools is something we're also starting to see. So it's still early days, but the data is highly suggestive that these tools are going to make a big difference in the world.
I think that's a great place to leave it as we bring this to a close. Mike, it's been a pleasure to have you on. So much insight shared today, and really thoroughly enjoyed listening to you. Excited for great things to come from Generate. Thanks so much.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.